Welcome to EDUCast, the podcast of the European Alliance EDUC. We are here to bring research to your ears. With this podcast series, you will get to meet several researchers from our partner universities and to discover exciting research and innovation topics, along with their links to society. Today, for this first episode, we are here at the University of Erwann with Assistant Professor Arthur David at the French School of Public Health, researcher at the ERSET, and Thomas Contini, PhD student at the University of Erwann and the ERSET lab. Arthur, Thomas, welcome.
Hello. Hello.
So, could you briefly present yourselves and your role at this university?
Yes, so I'm, as you said, assistant professor at the French School of Public Health. I'm also the deputy head of the Department for Environmental Health Sciences. And what I'm doing over there is I'm trying to understand how people are exposed to contaminants. So this is what we call human biomonitoring. We collect samples from people and we try to analyze them to understand their exposure to contaminants. And to do this, we are using different approaches, targeted where we look specifically at these contaminants, or in an untargeted way where we can really profile without any a priori their exposure.
Okay, thank you, Arthur. What about you, Thomas?
So first, thank you for the invitation. So me, I'm a PhD student, and I will spend the half of my PhD part in France and the other half in Maarten University. So I'm working on specific focus on placenta and on the exposure contaminants in placenta. So I'm trying to use, as Arthur said, untargeted methods mainly to study how contaminants can impact and also how can we found some metabolic change in placenta samples in order to adapt this to human cohort.
Okay, so you will spend, we were saying this but you will spend 18 months in France and then 18 months in Maarten University in Czech Republic. That's correct?
Yes, that's correct.
Okay,
that's
for your thesis.
That's it, yes.
And could you both tell us a little bit more about where you work? Because we have these preconceptions about what laboratories look like. So maybe if you could, describe a bit more what would be, I wouldn't call it your typical day in a lab, but labs can be very different, as we know, depending on what we are researching. So what is your day-to-day lab situation?
Maybe I can describe what ERSET is, because this is an incredible research institute. It's called the Research Institute for Environmental and Occupational Health. And we have many different teams, and basically we are studying the impact of environmental factors on human health. And we have teams working on this, and we have an analytical platform, and we are working in one of these analytical platforms called the LERES. And basically over there we have mass spectrometry that we are using. It's used to analyze contaminants, for instance, in water, in environmental matrices, but also in human. And we are doing all research in this lab. So we have laboratories for the sample preparations, for the mass spectrometry, and all the research we are doing this in collaboration with the epidemiologists, because we are part of the team called Life Course Epidemiology and Exposure Science for Environmental Health. And basically all the research we are doing, it's in association with epidemiologists. Like this, we can try to understand how people are exposed to contaminants and what is the link with some health outcomes. So this is really an exciting place to be, because you can find in the same institute epidemiologists, biologists, chemists, and you have many different specialties, and you can really make great collaborations with all these people.
Okay, that is a lot of people indeed. And what about you, Thomas? As a PhD student, what is your day-to-day situation, and how do you experience the lab, to say so?
Well, first, I totally agree with Arthur about this huge aspect of multidisciplinarity in research, and specifically in ERSET, and even in the team called Elixir. Concerning myself, I mainly spend a lot of time in a laboratory called LERES with sample preparation, analytical time with mass spectrometry and analytical chemistry, basically. and also a lot of time with computing, like bibliography research too. And we have to say we spend a lot of time also in front of our computer. It has to be said. So yes, it's such a good environment, but it's really different for all partners.
Okay, well, yeah, indeed. I didn't think I'm not an environmental researcher myself, but I did not expect that the computing tools would be of such importance. You were telling me a little earlier that you are using very expensive material too, very expensive tools, research tools, and this is quite common research overall, but it's always some surprising details, and because sometimes we think that research is just based on taking a look at the existing papers of our colleagues, which is not the case. And yes, go ahead, Arthur.
Maybe, I can add something about computing, because it became something very important even in analytical chemistry, because we are facing a huge challenge at the moment. That's why we are calling, the part of the research we are doing, the exposome. We are using this concept to try to understand how environmental factors, but all of them, are interacting and can impact our health. And when you focus specifically on the chemicals, you can call it a chemical exposome, because basically there are hundreds of thousands of chemicals present in our environment. And we know about maybe hundreds of them. So you can see that there is a big differences with what we can be exposed and what we know about them. So even in analytical chemistry, when you use high resolution mass spectrometry, for instance, then you have challenges to interpret the data. And the computing part is now something very important. So we are chemists, but we developed a software as well to help for the annotation of the signals that we are generating with these fancy instruments. So computing, it's now something very important in all parts of sciences and even in chemistry now.
Yes, I imagine it became more and more important in the last 3 decades with the development of all computing tools, in fact.
Yeah.
Okay, so Arthur, you told us about the exposome. I know that this is what you are currently working on. Maybe you could both tell us a little more about your current work. Maybe whether it's a big project or a smaller task, maybe something that is really, let's say so, taking most of your time in your mind. Could you tell us more about this?
Yes, of course. So the first step is to be able to have the right methods, and this is the big challenge we have, because we have these thousands of chemicals present in the environment, and we don't have the tools to analyze all of them. So what we are doing, a lot of our time is dedicated to method development to be sure that when you get a blood or urine sample, you can reanalyze everything that is in this sample. So we are developing new methods for the sample preparation, for the data treatment. So that's a huge part of our work. We developed, as I said earlier, a software called Xenoscreener. It's going to help us to be quicker in the way we can generate the results. But of course, that's only the first step. The first step is just to understand the exposure. And the second step will be able to try if there is some link between this exposure and health outcomes. And one of the big projects we have at the moment, it's called Eglantin. It's funded by the Fondation pour la Recherche Médicale in French. And basically, it will be using a huge cohort of 10,000 people. So we'll be using all the tools we've developed in this cohort, and the aim will be to understand the link between chemical exposure and neurodegenerative disease like Alzheimer's, for instance. So that's going to be really the second step, because for the last 5-6 years we developed the tools, and now we are happy because they are mature enough to be adapted to, let's say, large-scale epidemiological studies. So this is one of the first big steps we are taking into trying to understand the chemical exposome and how this chemical exposome, when I say chemical exposome, I mean all these chemicals present as a mixture in our body, and trying to understand how these chemical mixtures can be linked to disease like Alzheimer or other neurodegenerative disease.
And how does that impact your work, Thomas, for instance? I mean, how author's work maybe has a place in your work and future thesis? If it does, if there is a direct link, could you...
Yeah, obviously there is a direct link because all the preparation step to organize this PhD project is attributable to Archer and also over PhD student, postdoctorant of this team. So mainly the sample preparation. For example, for my placenta samples, I will use some sample preparation methods developed by Ragad Al-Salih, which was a former student in the team. And yes, also concerning, as Arthur said, the computing process for the pre-processing method to annotate compounds and to determine specifically which compound we can find in any sample. There is a huge development of this aspects in the team, which will benefit to my PhD project.
Okay.
And what can I just say to complete what Thomas says is
Sure.
that we are facing a huge challenge in terms of understanding this chemical exposure during the pregnancy time. The pregnancy time is a very sensitive period of time. It lasts 9 months. And it's very difficult for us to know what happened during the 9 months because it's very long and we don't have the methods to be able to monitor everything that happened during these 9 months. And this can have very dramatic effects when you are exposed to many, many chemicals because you can have effects directly at birth, but even later in life. So it's very important that we are able to monitor what happened during this pregnancy time. And Thomas, the PhD of Thomas, is really important from this point of view because he will try to see how the placenta, a very peculiar organ, can help us to understand what happened during these 9 months. So he will use this placenta and with this new method that we call, untargeted, he will see if there is some chemical mixtures accumulating during the long term, if there is some disruptions in some fundamental biological pathways. He will be able to see everything. And for that, first, you need to understand a little bit more about the placenta. So this is a very important subject into our work.
Mm. And I imagine this will make us able to solve some cases because I do recall some news that I may have seen on TV or in the papers where you see small villages or pockets of cases where children were born with different diseases, and we know that these territories were very polluted by chemicals, and there was no yet established direct link with these chemicals, but we know that probably these have an impact. So I imagine that your methods, the new methods you are bringing, will finally be, make us able to create a link between
these.
You are
completely right. We are really hoping that we will be able to understand a little bit more with this research and these tools that we are developing. This is a very important point you mentioned here.
And maybe a small question is, how did you become interested in this topic? What led you, maybe with your educational background, to go towards this topic and led you here?
So, concerning myself, I always be interested by biomonitoring and to understand what is the link between a chemical fact or an exposure fact and its consequence on life. I'm not totally focusing on human, but also as animals too. It's more difficult to have information for animals because it's not as popular as human. But yes, that's the point for me.
Okay. What about you, Arthur?
Science has always been something very important in my life. I really like the natural world, and I started my research studying mussels, then fish, then bees. And then naturally, I become involved with human research, but everything is just one thing actually, because when you are studying bees and what they are exposed to and the decline about bees, you can see that it can impact indirectly also humans. So even when you study the environment and see how it can be impacted by human activity, you are actually studying as well human activities. So it just being part of my life since I was a child, being involved, interested, I would say, in science. So it was quite an evidence to become a researcher. I didn't have other choice, I guess.
Okay. I would like to talk a little more about the European aspects of what you are working on. I know, Thomas, that you have this partnership for your thesis between France and Czech Republic. So before talking a little more about this, I would like a very quick sentence on your view and your opinion on the world of research throughout the European Union and beyond, maybe, as of today.
Well, Europe is now so important in research. I mean, we are at the same stage that the Human Genome Project was, at the end of the 90s, and now we are studying the exposome, and we need clearly the same kind of international collaborations. That's for sure, and that can happen only if we do it, as a group. And the Europe is very important because there have been a lot of funding recently to study the exposome, to fund big consortiums of research, but also to fund, for instance, research infrastructure. So we are involved in this kind of big project in Europe. HPM4U, Park, and now IRIN, which is a research infrastructure. So I would say that's the only way to really overcome some of the challenges that we are facing.
And what about you, Thomas? As a PhD student, what's your view on research at the scale of Europe or beyond, international research?
Well, I think it's a very good point to have some exchanges between countries, to have mobility possibilities, to make, for example, like 3 or 4 months in another lab, to try to use other techniques and new methodology and also new thinking way. So yes, I think it's a very interesting part.
Yeah. I completely get what you're trying to say, and also we're also here thanks to the EDUC Alliance, and I wanted to talk a little more about this. Can you tell us more about your concrete and direct links with us, the EDUC Alliance, as of today?
Yes, we were lucky enough to get a funding from EDUC. Actually, this is for the PhD of the funding for Thomas. And this is in collaboration with Masaryk University and more specifically with Resetox and Jana Klanova and Elliot Price. So this is a collaboration, meaning that, as you said, there will be 18 months funded in France and 18 months in Resetox. And this is really a good opportunity because we have really complementary expertise skills, meaning that Thomas will be able to benefit from the skills we have here, the specific skills, and the ones from Resetox. So this is really, we hope it's going to make the PhD of Thomas much better thanks to that.
So if I'm right, Thomas, all what you were mentioning earlier will be part of this partnership. All your work on the placenta and the study of it and the chemical interactions, all of this is part of the partnership.
Yeah, absolutely, yes. Okay. It's part of the partnership, and yes, as said earlier, the main thing is that I will do other methods in Czech Republic than in REN, and it's a complementary approach.
So will there be some sort of comparative approach as well to see how each method works differently, and maybe if there are some results that you may be able to crisscross, to say so?
Yes, absolutely. It's we want to merge, we will use basically 2 methods with liquid chromatography and gas chromatography with high resolution mass spectrometry. So the 2 methods doesn't cover the same chemical range of products. And we want to use the 2 methods in order to merge results, and the aim is also to create a big human placenta database concerning all chemicals we can find in placenta.
Okay, well that's quite impressive, I must say. Before ending this podcast, because we're almost at an end here, I would like to know if there are 1 or 2 research publications which you read recently that you would recommend. There may be the ones from colleagues or anything related to your topic and beyond.
So maybe not publications, but as a matter of fact, we organized an Exposome summer school 2 weeks ago, and we invited many researchers. And what was really interesting is to have point of view from researchers that are completely outside of our field. So we invited, for instance, a philosopher of science called Elodie Giroux, and she really helped us to be more open about how we should design our studies. And there was Paolo Vinis, so we recommend if you don't know his work to read it so that you can really be more involved in exposomics research.
What about you, Thomas? Any latest publication that you appreciated?
Well, I will talk about a publication which is mainly in my subject, which concerns placenta. And as I see, it's a Floridian researcher, which is talking about the difference of metabolome, such as all the little molecules we can find in different parts of the placenta. So it's a very interesting work. And for a more global thing, I will not talk about an article, but I will say to read the GHEC, for example.
Very, very interesting indeed, and quite precious in these times. Okay, maybe in a short line, how can people find out more about your work and keep up to date with news from your research laboratories and establishments?
Well, the ERCET Institute and EHESP, the French School of Public Health, have their website, and you can find many information on this website about our work on the exposome in general, or about the work of ERCET and the French School of Public Health.
Okay. Well, this episode is reaching to its end. Arthur, Thomas, thank you very much for coming. Do you have one last word for research, I would say?
No, but thank you very much for the invitation. It was nice to talk about this EDUC program.
Thank you very much for the invitation. It was a great pleasure.
Well, thank you again. And for the audience out there, well, see you in the next episode for another topic on research.